Being here. Thank you for giving the opportunity for me to tell you about my research. The title of my talk is Predicting Catalytic Functions with a Probabilistic Model for Genomic Context Analysis. This talk is 20 minutes, even though the schedule says 10 minutes.
It is 20 minutes.
So my name is Jeffrey Yunus. I am a PhD candidate in the UC Berkeley UCSF Bioengineering program and a member of Patsy Babbitt's lab at UCSF. So in the Babbitt lab, we collect large numbers of, or we look at, we deeply study, select mechanistically diverse superfamilies, and then we carefully look at the literature. We build protein similarity networks, sequence similarity networks. We look at structures, we build alignments, and we do all this in order to draw functional boundaries and to explore and to identify regions of the space that have not yet been explored. As you know, however, there's not very much experimental evidence in order to draw these functional boundaries. And the evidence that we do have is sparse and biased and only partially labeled, etc. So my project was to very carefully incorporate one additional piece of information to help refine these functional boundaries. That piece of information is genomic context. An example of manual genomic context analysis that inspired my work is by Frank Rochel's lab at Texas A&M. And this is a graphical representation of it. Here we have a query protein, which is unannotated. We have a few annotated proteins as deaminase nucleotide deaminases. And these edges represent sequence similarity, meaning that most of these proteins, this is not protein interaction network, most of these proteins are in different species. So because the query was only homologous to a bunch of sequences annotated as nucleotide deaminase, the query was a putative nucleotide deaminase. But when they looked at the genomic context, they saw that the query protein, their protein of interest, was surrounded by proteins involved in the histidine degradation pathway. And they, therefore, they didn't think it was very likely that it was actually a nucleotide deaminase. They hypothesized it because histidine is not a nucleotide. So they hypothesized that maybe it was actually, by looking at the substrate substructures, they hypothesized that maybe it was actually a N-formamino L-glutamate deaminase, and they validated this experimentally. Now, while this is good research, you can imagine a case where we have a query and none of the homologs are characterized and none of the members of the pathway are characterized. And you might initially think that there's nothing we can do to get any information about the query, but if we expand this context, then you might be able to start thinking that you can come up with some explanation about how you can propagate things a little bit and come up with some explanation for what's going on in this network to glean some information about the likely, some likely functions for the query. Now, that sounds good, but when you look at the actual scale of this problem, it's much, much greater. And it's clear that manual genomic context analysis at this scale is infeasible. So the question we're trying to solve is how can we automate genomic context analysis? But the first steps to do this, or I guess the perspective that we're going to take on this, is how can we make a framework for protein function prediction that can use biological process annotations and functional association data. So we take a probabilistic approach to solve this problem. A probability model is well suited for protein function. It's good at dealing with sparse, noisy data. It's good for dealing with function propagation, which is also noisy, and for doing things like quantifying the uncertainty in the results. And if we can calculate the joint probability of a network, and by that I mean the probability of a given functional assignment to all the proteins in the network, then we can really calculate anything we want by marginalizing out and conditioning. In other words, we can ask what's the probability of a query having this N-formamino L-glutamate deaminase activity, or we can even calculate cool things like the variance or uncertainty in the model to help us with target selection, which is trying to find out which proteins we should subject to further study. The framework I use to solve this is probabilistic graphical models. It has been successfully used for protein function prediction with several different models of it. Here are some of the seminal papers in the field. It's particularly appropriate for protein function prediction because probabilistic graphical models are good for structured and relational data. Namely, it's good for semi-supervised methods. As you know, we talk a lot about how it's such a liability that we have so many unannotated proteins, but a semi-supervised method actually uses that as an asset and actually can use those unlabeled proteins to help us draw functional boundaries. And it's also good for structured labels, both inputs and outputs, and ones that are only partially labeled as inputs. So anyway, this right here is saying that the model is going to be a product of local functions or of functions over subsets of variables and then normalized in order to be a valid probability distribution. So if we can choose what these smaller components are and learn the parameters, then we can make some progress, hopefully. So the questions are, what are the smaller components of the model? So let's start off by introducing a very small protein network of 3 proteins. Here we have protein A and B are similar in sequence. Protein B and C are functionally associated. According to string, it might mean they have the same, they're in the same genomic context or co-expression data, something like that, or phylogenetic profiles, et cetera. So to begin the model, we model a protein's molecular function with a hierarchy of Bernoulli random variables that looks a lot like the gene ontology. They each take a value of 0 or 1. There's several reasons for doing this. Compared to a multinomial, we can represent that a protein has, or we can predict that a protein has multiple functions at once. It can naturally do structured outputs as well as structured inputs with partial labels. And we can model, for each of these variables, these Bernoulli random variables, we can model dependencies with other variables in the network. When 2 proteins are functionally associated, when 2 proteins are similar in sequence, we connect the corresponding terms in molecular function. So what this does is, in terms of that probability model that you saw at the beginning, this will add a factor that says 2 proteins that are similar in sequence are likely to have similar functions, as long as that's what the parameters learn. This part of the model has many similarities to this Mitrofanova paper down here, but there's also several important differences as well. Now, when 2 proteins are functionally associated, when 2 proteins are functionally associated, then we model biological process in an analogous way as molecular function. So we're modeling multiple aspects of protein function, and then we link the corresponding biological process terms. So normally when we integrate networks, or when we integrate multiple sources of data, we might just say across a sequence similarity edge, we link the molecular functions, and across a functional association edge, we link the molecular functions also. And that doesn't seem like it's the best use of data. Because it's kind of making the assumption almost that when 2 proteins are in the same pathway, they're likely to have the same molecular function. So right now we have just a bunch of, as you can see, these are not connected. We have a bunch of independent networks. And to complete the model, we add interontological edges that represent the best correlations between molecular function and biological process. So you can picture this complete model as belief about function propagating around the network, but in a probabilistically rigorous way. The parameters of a directed graphical model, a BayesNet, the parameters are functions that have a global probabilistic interpretation. That's unlike a Markov random field where you have to normalize it and it really depends on the structure of the network. Namely, this is the probability of the variable given the parents of the variable. Well, the network I just showed you, while a gene ontology is directed and acyclic, the protein network is definitely not directed and acyclic. And the protein template might not, when you add the correlations between multiple ontologies, is also neither directed or acyclic. So to address this and give the model directionality, first we take a minimum spanning tree of the protein network, we route it to a query, we print it to the evidence, and we direct everything outward. And then we end up with a parameterization, which could be a maximum likelihood parameterization that has a global probabilistic interpretation for the model that I just talked about. It's the probability of a protein I having go term T given its go term parents for that same protein and its protein network parents for that corresponding term. And that might be, in this case, blast, string, or for the interontological edges, it depends. Its parent, one of its parents, is the interontological parent of that go term, but in the same protein. So you can see that here. So here's a term, and its parents are here and here. Over here, its parents are this person and this variable also. So now that we know what these are, we can just count them by looking at Goa. So the idea is just to look at neighboring annotated proteins in Goa and count them. But since missing data will result in a very high likelihood of gaining a function or losing a function, we weight the instances by their information content when counting them up. And that seemed to help things quite a bit. So parameter slides are pretty... Oh, one thing that's kind of novel here that I should point out is also that we learn parameters for individual Go terms, for each individual Go term. So this might be the first case where we have parameters that say the likelihood of gaining or losing catalytic function activity term is very low, but the probability of changing substrate could be significantly higher. So parameter slides are usually kind of dense. To summarize it in this kind of flow chart, the general idea is to learn the parameters, and then for each query protein, build a network. We do that by collecting the query homologs by BLAST, collecting functionally associated proteins in string, and then running diamond for an all-by-all BLAST. Building the probabilistic graphical model, that means getting that directed model that I just talked about, determining the candidate functions, then making one factor for which is the probability of T given the parents of T for each protein I in term J. Run inference. There's a number of ways to run inference on a probabilistic graphical model. And finally, output the results. In terms of results, they look like this. This is visualized in Cytoscape. It has a nice intuitive interpretation. Obviously, these are proteins. This is from the perspective of a single Go term. The query is right here. These four proteins had positive evidence for the term that we're looking at. This one had negative evidence for the term that we're considering here, the Go term that we're considering here. And the cluster, the homologous cluster, the query, there's a cluster of homologs that is homologous to the query. That has some evidence, but it does not seem to be directly related to the Go term that we're looking at. After running inference using this method, you can see that we have a certain prediction for the query for having this Go term, and you can see everything else propagated as you would. It looks reasonable. Keeping in mind that we did not just run this method for a single Go term. This is a structured output. We were predicting everything for every relevant Go term that we have. In this specific case, behind the scenes, this was our molecular function structure, our aspect that we were modeling. This is the biological process. This is the biological process that we were modeling. And if you zoom in, you can see that we had a certain prediction for this guy and less so for these two. So I really like the evaluation work that has come out of CAFA. I insist on doing this CAFA-like evaluation, leave most recent out. We have a lot of names for it. But the idea is leave out and predict on the most recent proteins. Just for my own interest, I'm looking at I'm looking at catalytic annotations, but that was fairly arbitrary. Things that I'm not filtering out include I'm looking at all Go terms, even if they only have one associated protein. So I don't set a minimum number of associated proteins before I'm required to predict that term. And I don't filter out any proteins based on the number of labels it had in the annotations it had in the training set. So this is what we've seen a few times before today. I think it's always woefully incomplete. Usually around here, you realize you have to recompute your results for some reason, and then the cluster goes down here for a kernel upgrade. Internet goes out, and then this is when you write to the CAFA organizers begging to let you submit your predictions. The metrics I use for evaluation, again, out of CAFA and come from Wyatt Clark and Paige's lab. I think these are phenomenal contributions, and I always talk about these when presenting. If we want to compare, see if we got the right label, comparing using equality is a terrible idea because the true annotation, the true label has 50,000 terms and it's incomplete. So there's no way that testing for equality is going to work. All methods are just going to get zeros. So I use this method that is the information. So a better approach would be to use precision and recall, but these are information content versions of precision and recall. So the idea is remaining uncertainty is the sum of the information content of what's in the true but not in the predicted, and the misinformation is the sum of the information content of what's in the predicted but not in the true, and then you can weight these to evaluate entire methods different ways, either to get normalized versions and weighted versions. So this is the best stuff over here that we're trying for. I have to speed up a little bit, but this is very, very, very encouraging, I think. BLAST is this. BLAST is a method that is collected homologs at 1 e to the negative 12, and then it did something like CAFA, except it's using bit score per residue in order to kind of to look at whole alignment scores. And you can see that around like 60% of the average remaining information content collected, it's doing, looks like it's getting like half of the error in terms of the weighted information content of mispredicted terms. Also encouraging is that if you don't model multiple aspects of protein function, then the performance can actually degrade. And that's what several methods do. But it is still a work in progress. Here we have the full method modeling biological process with different numbers of interontological parents. And you can see it improved it, and it improved it from the previous line I just showed you, but not as well as the fusion with sequence similarity and not modeling biological process. So still a work in progress, but certainly a lot of promising things. Future directions, certainly the most straightforward extension is to also model cellular component. We know cellular component is high throughput and somewhat unbiased, and we know that if there's some enzyme that's happening outside the cell, then it really changes our posterior probabilities of what that molecular function might be. We've talked a lot about this in this conference, but the whole point of this method is to really have finer grained similarity measures and propagate only the respective or the appropriate aspects of protein function. So if we can narrow this down even further into domains or regions, then that would be great. I'm very excited about the prospect of using substrate substructure and that stuff, and I mentioned target selection already. Finally, I'd like to acknowledge my advisor, Patsy Babbitt. She's a phenomenal advisor and a very skilled bioinformatician. She's retiring, so that'll be a loss to the community, but we're all happy for her future. several members of the lab that helped me a lot, my advisors, and several people who have helped me both at UCSF and outside UCSF. I'd like to thank funding from my program for coming, as well as a generous grant travel award from the NSF. Thank you. I really do hope to hear some feedback outside. You can always email me, and thank you.
Thank you, Jeffrey. Okay, in the interest of time, we won't have questions.